Hi, my name is Taylor Albrecht. I'm a senior in the MSTC program at Paul Laurence Dunbar High School, and for my capstone research project, I worked under the mentorship of Dr. Sarah DeRazio from the Microbiology, Immunology, and Molecular Genetics Department at the University of Kentucky. And my research is titled Comparative Genomics and Analysis of Neurotropic Strains of Listeria monocytogenes. Before I begin my research presentation, I'm going to give you a brief overview about what I'm going to be talking about. I'll start off with an introduction, and then I'll go into my research goals and hypotheses. And for this research, I had 3 different projects, so I'm going to talk about each of them by methodology and results. I'll first talk about my MLST project, and then I'll go into INLF detection, and then I'll finish off with my genome completion project. After that, I'll go over the discussion and conclusions for each of those projects, as well as some further research that I have in my acknowledgements and references. And finally, I'll open the floor to questions. For this research, I was working with Listeria monocytogenes. Listeria is an invasive type of bacteria that most commonly causes infections amongst immunocompromised patients. Certain strains, however, called neurotropic strains, can inflict rhombencephalitis, and rhombencephalitis is an infection of the brainstem. What's unique about rhombencephalitis is that it can be extremely dangerous and it can infect healthy patients, leaving them with persisting and lasting neurological deficits. Currently, there's a big gap in understanding of neurotropism and how rhombencephalitis works, and it could also suggest that Listeria infections are more dependent upon the strain than upon the patient. There are also 4 known evolutionary lineages of Listeria: 1, 2, 3, and 4. Lineage 1 and 2 are considered to be the most significant and they're the most prevalent. Lineage 1 is typically found in human isolates, and lineage 2 is typically found in foodborne isolates. And a majority of the Listeria research is focused directly on those 2 lineages. There's also lineage 3, which is thought to be far less significant, and it's a little bit more rare, being only found in ruminants or non-primate animals. And lineage 4 is extremely rare and highly underrepresented. So what my research is focusing on is kind of making the connection between lineages and neurotropism, as well as just understanding how neurotropism works as a whole. My main research goal was to analyze and compare neurotropic strains of Listeria monocytogenes through the use of multi-locus sequence typing, or MLST analysis, which gives me access to a lot of genetic information about Listeria, like its evolutionary lineages, as well as clonal complexes and sequence types, and as well as gene investigation, where I was analyzing 2 neurotropic strains for the existence of a virulence gene called INLF, as well as genome assembly and completion of 2 neurotropic strains of Listeria. For my hypothesis for each of these research projects, for MLST analysis, I predicted that all of the strains were likely to be lineage 1 or 2, simply because those are the most common lineages, and so it's likely that it would be one of those 2. For INLF detection, I predicted that INLF does not exist within lineage 3 strains of Listeria. And for genome completion, I predicted that INLF will not be found anywhere else within the genome. Before I go into talking about each of my individual projects, I want to go over each of the strains that I was working with for this. The first 2 here are EGDE and EGDE INLAM. EGDE is the most common strain of Listeria. There's been a lot of research done on it, and it's kind of the baseline for a lot of Listeria research. So INLAM is a genetic variant of EGDE, and so they have the exact same genetic composition, except for the fact that INLAM is lacking the gene INLA. I also worked with UKVDL4, 7, and 9, which were all isolated by the UK Veterinary Diagnostic Labs. And I also worked with 2010L2198, which was isolated by the CDC. Now, the 2 most important strains in this table are UKVDL9 and 2010L2198. That's because both of them are neurotropic strains, and so a lot of my research is focused on those 2 specifically. Now I'm going to go into my first project, which was MLST analysis. For this, I performed multi-locus sequence typing on 6 different strains of Listeria. And the basic procedure of this was performing a polymerase chain reaction or PCR reaction on each of these strains. And I did PCR for 7 different housekeeping genes, which amplified the DNA of the specific gene. After that, I performed a gel electrophoresis to confirm that there was a DNA product, and then I cleaned up the sample and sent it off to be sequenced, and after processing that sequence, I was able to submit the sequence of each gene to the MLST past year database, where each gene sequence would be assigned an allele number, and after compiling all of the allele numbers for a specific strain, I was able to receive the lineage, clonal complex, and sequence type of each of those strains. The results for my MLST project are shown here in this table. For EGDE and INLAM, they both had the same genetic composition and they were both found to be lineage 2. And surprisingly, all 4 of my other strains were found to be lineage 3. This was extremely shocking considering lineage 3 is thought to be so rare and insignificant, especially when considering the fact that 2 of those strains were neurotropic. And so that caused the rest of my research to focus directly on 2010 L2198 and UKVDL9 specifically to see what makes them neurotropic. And this leads me into my second project, INLF detection. And for this, I was analyzing UKVDL9 and 2010 L2198 for the existence of a virulence gene. And a virulence gene is a specific strain in Listeria that codes for a protein that allows it to cross the blood-brain barrier. And the first group of genes that I was looking at for this were internalins, which is a family of surface-binding proteins. So there's a lot of potential for Listeria, or an internalin granting Listeria the ability to cross the blood-brain barrier and have access to infect the brain. The specific gene that I was looking at first is called INLF, and the reason I was looking at INLF is because a couple of research papers had suggested it as a virulence gene, so I wanted to prove that theory. And in order to see if INLF was a virulence gene, I first had to see if it existed within either of my 2 strains. Pictured here is a diagram showing an example of the top one, a lineage 2 strain, and the bottom, a lineage 3 strain. As you can see, EGDE, or the lineage 2 strain on top, contains INLF, and the lineage 3 strain does not. However, the 2 genes surrounding INLF are conserved within both of the lineages. So what I did is designed a primer that amplified the region containing INLF to see if it existed within my 2 strains. If INLF was there, it would have had a 3,000 base pair product. If it was not, there would have been a 340 base pair product. The results for the INLF PCR shown here. Lane 1 is just a base pair ladder that shows me where I am numerically. Lane 2 is EGDE with a 3,000 base pair product. And lane 3 and 4 are UKVDL9 and 2010 L2198 respectively, each with a 340 base pair product. And lane 5 is just another base pair ladder. So EGDE as a 3,000 base pair product, meaning it does contain INLF, and the other 2 strains have a 340 base pair product, meaning that it does not contain INLF. However, that does not guarantee that INLF doesn't exist anywhere else within the genome for UKVDL9 or 2010 L2198, so that led me to my third and final project of genome completion. And for this, I was working to complete the genomes of UKVDL9 and 2010 L2198, and I used 2 different approaches for each of those. For UKVDL9, all I did was send it off to ACGT Inc. to be sequenced, and they returned to me a completed genome that was sequenced and annotated fully. However, for 2010 L2198, the process was a little bit different. There was already a pre-existing draft genome that was created by the CDC, so it was my job to go through and fill in the gaps of that draft genome. And shown here is a diagram that depicts the basic sequencing process. It begins by cutting up the genome into millions and millions of sequence reads, which are about approximately 150 base pair lengths of DNA. After that, all of the reads are sequenced and aligned to form larger groups of DNA called contiguous sequences, or contigs. And once all of these contigs are created and aligned in order, there are gaps existing between them. And once it's at this stage, it's called a draft genome. And my job now is to fill in those gaps that exist between all of the contigs. I used a similar PCR design that I did for the INLF detection project, and I was able to design primers that amplified the regions containing the gaps. For the genome completion results, the PCR sequencing method has proven successful. I've been able to amplify a lot of the gaps and get those sequenced and added to the genome. However, due to the COVID-19 outbreak, I was not able to return to my lab and complete this research, but I have been able to design 23 sets of primers, and at this point, I just need to be able to get back into the lab to test those and finish up this project. However, preliminary research has shown that INLF does not exist within the genome of UKVDL9 or 2010L2198. Now I'm going to go on to my discussions and conclusions for each of my projects. For MLST, it was very surprising that all experimental strains were found to be lineage 3, especially with UKVDL9 and 2010L2198. This really expands the scope of the relationships between lineages and neurotropism, and I think that now that there's 2 lineage 3 strains that were neurotropic, there's a lot more room for performing research on that relationship between neurotropism and lineages. And I think it also expands that significance of lineage 3 strains, and so hopefully in the future a lot more research will be done on those to analyze them a little bit more thoroughly. And for INLF investigation, I found that the INLF gene does not exist within the expected area of UKVDL9 or 2010L2198. That in turn means that INLF is not a virulence factor for neurotropic strains of Listeria that are of type lineage 3. However, there is room for potential error because it could have existed in any location outside of that expected region just due to genetic variance. So that again led me to my third and final project, genome completion, where I was able to confirm that INLF or a similar gene did not exist within either genome. So that essentially confirms the conclusions of my INLF detection project, meaning that INLF is not a virulence gene for lineage 3 strains of neurotropic Listeria. However, just because the neurotropism is fairly unknown, I think that a lot more research needs to be done on neurotropism as a whole just to explore it and learn a lot more about it. For future research, I would like to complete the 2010L2198 genome and compare it to that of UKVDL9 just to see what are different and similar about them and what genes they may have in common. I would also like to perform a genomic comparison of the genomic composition of different lineages as well as non-neurotropic strains versus neurotropic strains to see, maybe point us in the direction of other virulence genes as well. And I would also like to continue the search for virulence genes in the future. For my acknowledgements, I would like to thank Dr. Sarah D'Orazio, my mentor, for providing me with these resources to perform this research, as well as guiding me along through the whole process. I would also like to thank the members of the D'Orazio lab, Juyong Cho, Taylor Sine, and Katie Alexander, for helping me with the little things in the lab and teaching me all of the tips and tricks about working in a lab. I would also like to thank the Microbiology, Immunology, and Molecular Genetics Department of the University of Kentucky, as well as Kentucky Young Researchers Program, and Mrs. Karen Young and the Math, Science, and Technology Center for providing me with this wonderful experience and allowing me to gain research experience as just a high schooler. And I would also like to thank my friends and family for guiding me along the way. And these are my references. And now I would like to open the floor to questions. Since this is virtual, I left you my email. Please feel free to contact me with any questions at taylor.albright@stu.fayette.kyschools.us. And thank you for listening.